It's a great pleasure today to introduce Cody FitzGerald as our seminar speaker. Cody got his PhD in the University of Utah in mathematics, and since then has been an independent fellow at Northwestern, first at the Quantitative Biology Center that predated NITMB, and now as a James S. McDonnell Foundation fellow, and has been doing a lot of great work on trying to figure out how to use biological data in a data-driven way. So, thank you.
Okay, thanks. Okay, so today I'm going to tell you about is it okay? The audio is okay? Okay. Today I'm going to tell you about a few projects that I've been working on. Okay, great. So first I'm going to tell you a little bit about my general interests, then I'll tell you about an ecological, epidemiological project, then a project looking at gene regulatory network inference, and then a physiology project, and then just very briefly like newer projects. And how I've kind of organized things, right, is this is going to be like very little data in terms of the first project. Then we're going to move into like more data, and then even more data, and then just a huge amount of data, right? So we're going to kind of like go through. Yeah, so that's going to be kind of the trajectory here. So in terms of my general interests, right, I have a lot of organismal biology interests, right? So I'm very interested in dormancy behaviors and processes, right? So these are things like hibernation, viral latency, the dormancies of plants, right, and how that might relate to human disease. Then I'm very interested in kind of multi-scale complex systems, control of ecological and human systems, and then applied math, data-driven modeling, optimization, model selection. Okay, and then I just wanted to flag that I'm organizing this workshop here, right, and it looks at kind of multi-scale biological adaptation in changing environments. And so it's going to be next fall. And I just wanted to kind of flag, like we have like a broad range of organizers. So we have some math people, like me and Alan, but then we also have people studying avian physiology, right? Like Derek and hibernation biologists and the neurobiologists, and some other people as well. So kind of should be kind of a cool thing and bring together like a really different groups of people that don't normally interact. So I'm very excited about it. Okay, great. Okay, so the first project I'm going to tell you about was it's a project I did with Jim Keener at Utah. And we had been thinking about this for a while, but we recently wrapped it up. And so it's kind of an interesting like bifurcation story and things like that. But the real reason I want to talk to you about it is more about a problem with parameters that we're going to get into. Okay, so mostly in the Western US, there's this big problem right now with this disease called chronic wasting disease. And you can see here, right, we've got a deer infected with this disease. And you can see it has kind of a sickly look. It exhibits kind of odd cognitive processing. And yeah, it's a bit weaker, right? It's going to walk in a non-optimal way. And then it's a very long disease progression, something like 2 or 3 years. And it's caused by basically a misfolded protein in the environment, right? And these prions, they have a super long half-life. So they just sit in the environment for basically decades. And there's no clear way to eradicate them, right? So there are other similar problems with things like scrapie in sheep. And what people have tried to do is just like burn the entire field where the sheep were living. And even that kind of intervention will not impact the infectivity of these prions. Okay, so it's very widespread, right? And there's no vaccine, there's no way to kind of decontaminate the environment, right? So the question we wanted to ask is, right, what if you just kind of introduce wolves into the environment? Would that help to control the disease, right? And let me just think if there's anything else I should say here, right? How do the deer actually get the disease, right? It can be direct, right? One deer interacts with a sick deer, right? They can contract it that way. But the challenge is that you can also pick it up from the environment. And so, maybe you're a deer and you're trying to eat and you're interacting with it in the soil or the water, So you have both direct and indirect transmission of the disease. Okay, and just to give you a sense of like how bad things are, this is a survey from Wyoming, and what they're doing is they have widespread hunting, but they have a testing program. So they're taking brain samples from elk and deer that are harvested by hunters, and you can see that here, right, you're just saying like, okay, out of all the deer that were killed by the hunters, how many of them had disease? Right, and some regions are like pretty good, right? You have maybe like 0 to 5%, whereas some regions are 60 or 70% prevalent. So it's pretty serious. And then maybe the other challenge with this kind of thing, right, is I'm not an expert on epidemiology or something like that, but maybe the thing you don't want is like people ingesting these prions and then them kind of evolving in like a human body. Like that's because you get a risk for jumping. Okay, so we wanted to analyze kind of a mathematical model of the system, and just kind of do like a classical bifurcation analysis and control of the system, right? So I'm tracking kind of density of susceptible deer, density of infected deer, and then the density of the prion in the environment, right? And so my wolf population is going to be constant, and it's going to come through here in these rho SW and then rho IW, okay? And so what we're going to do is we're going to just do the deterministic analysis of the model, right? And then we're going to stochastically simulate the system, and then I'm going to ramp up predation by wolves, right? And just see what happens in that case, right? But the real reason I want to like bring this up is we're going to have what's called a structural identifiability issue in the model. And so that's going to kind of bleed into some of the other projects I talked about, right? So to do the analysis, we had to come up with some parameter values, right? And some of them are pretty easy to come up with, right? So I was emailing all these biologists and talking to them. And, basically like things like R, that's like pretty easy to know, but, and maybe we know something about the time scale of the degradation of the prion in the environment, but parameters like gamma E and epsilon, those are very difficult parameters to estimate, as we'll see, right? And so I was finding these biologists, and they're telling me things like, oh, those actually depend on, the species of deer, and it also depends on the environment, and it also depends on like many other factors, and then things get really bad because these prions are actually like found in ticks and insects, and they also are inside plants, and if the deer eat those, then right. So there's a lot of things that are going to be coming that are challenging here.
Question.
Yeah.
So like, can you give an intuition why like adding predation might help control the disease?
Yes, we'll get there, right? But the general answer, right, is like they're going to kill the sick deer, right? Because they're just going to.
So it's like easier to kill the sick deer.
Right. Right, because they're moving more slowly, right? So there's a relationship in the model between like rho S and rho I, right? But there's also another reason that we'll get to, right? So if I do the bifurcation analysis, right, and here, right, this is where the locus of Hopf points in the system are, right? And we can do an analytic calculation in terms of just the model structure, right? So here, right, we see these qualitatively different regions. So here I have this predation of infected deer, and then here I have the carrying capacity of the system, right? And I see I have a region where I have no disease, then I have endemic disease, and then I have oscillatory outbreaks of the disease, right? And we have analytic solutions for the transitions in the system, right? And here I'm just plotting in this 2D space, but we have the whole surface of where it occurs in the model.
Okay.
Yeah.
So when there's no disease, is one extreme of this that there's just no deer?
Yeah, exactly. We're not showing it on this slide, but yeah, it does. The system does have an extinction state, right? Okay, so here I'm showing you the deterministic solution in the dashed line, and then we have a stochastic realization of the system in the solid line, right? Okay, and so like we simulate the system treating deer as particles, right? And we use the Gillespie algorithm, right? The kind of classical chemical reaction exact simulation. But one tricky part that we had to modify, right, is E is continuous, right? So there's a little bit of a tweak we had to do there. So that's in the paper. But what you can see is if I have no wolves, right, a lot of the trajectories of E start to kick up, right? I get the infection spreading, but some of them don't, right? And then as I'm just going to ramp up E as I go, right? And I can start to see, okay, my red line, right, which is the infected deer, right, like that's pushing down. And so that's a good sign, right? And I'm going to increase even further, right? And I'm able to push the system, right? I'm pushing the amount of infected deer down, and then the infected or the non-infected deer, right? I'm getting stable population, right? Okay, but the problem is, right, we're enacting these controls at time equals 0, right? But it is not time equals 0, right? Things are widely spread already.
Sorry, Cody, even before you get to this, I don't think I really understand what's happening. So you mentioned this unidentifiability challenge.
Yep.
So when you make these plots, do they correspond to just one possible solution out of all the possible ones, or what are we looking at?
So here, right, I identify the regions of the model, right? So here, I can say generally what qualitative behaviors will occur, right, in different regions of the parameter space, right? And I have analytic solutions to those, right? So I can say, okay, here's my surface of where oscillatory behaviors occur in the model.
Okay. But I mean, there's lots of parameters here, right? And you're making these 2D plots. So I still don't quite.
So we're fixing the parameters. We're fixing the parameters. The results we show are for specific parameter values, right? And in particular, where the model oscillates, right? But one thing we do in the paper is we say, okay, like let's be careful when we're estimating parameters from the data, right? And so there's kind of two things happening, right? We do kind of a traditional analysis, right? And then we're pointing out this problem, right? Because we're not interacting with, like, I'm not a manager of a wildlife population, but some people are, and they're using these populations or the parameters, right? So I'm picking some parameter values to get the results I'm showing you, and then I'm flagging an issue of if you want to use these for management solutions, we need to be careful about this.
Okay.
I had a quick question.
Yeah.
So when you say that, sorry, could you go to the next slide?
Yeah.
So when you say that there are like surfaces which, like, demarcate like the regions 1, 2, and 3, like, what do you mean by a surface? Like, in which variables are you using?
Right. So here, this is
Oh,
the expression itself, right?
I see.
And I can do a similar thing for the transition between, state 1 and 2. Yeah.
Can you go back to your bifurcation, your equation?
Yeah.
Which parameters are unidentifiable, and are they in this equation?
Yep. So the unidentifiable parameters are going to be gamma E and then epsilon, right? And so what we do is we're just going to arbitrarily fix one.
Okay, so gamma E, and do they appear separately, or it looks like they appear together?
When I non-dimensionalize the system, they will always appear together.
Okay.
Yeah.
And if you look at gamma E and epsilon together as the product, since they're appearing together in your bifurcation curve, is that identifiable?
Yeah, that
the
is
product is
identifiable.
identifiable. Okay,
Yeah,
so the
the product.
bifurcation curve actually is identifiable because epsilon and gamma E only
That's
appear
right.
together. Okay,
Yeah.
cool. Thanks.
Okay, so we have delayed control here. And then so what we're going to do is we're going to just, instead of introducing wolves at time equals 0, right, I'm going to delay, right? And I'm going to make it basically the wildlife managers will introduce wolves at a certain prevalence condition, right? And so what you can see, right, is we're saying, like, let's wait until the infected population reaches 1/20th of the carrying capacity. And can you still get it to work? And you can, right? So you can see qualitatively, right, I'm driving the infection down in terms of S here is, right, the number of susceptible deer, and then here I have the number of infected deer, and then I'm pushing it down, right? Okay, so to the question we had, right,
Sorry, a question.
yeah.
Like on your plot, I'm wondering like if you ever get like extinction events, because it's kind of quite easy to get them? Or
Mm.
like,
Right. Of course, if I make wolves, if I just increasingly add more and more wolves, right, it would drive the system to extinction, right?
like these are like kind of stable.
Yeah. Okay. Right. So I have there are two benefits, right? One is that it controls the infected population, right? So I'm not going to get right as much direct transmission, and I'm going to prevent like these formations of the environmental reservoirs, right? But it also controls the size of the susceptible population. So if I imagine I have just a susceptible population, but I have the prion floating in the environment, right? The bigger the size of the susceptible population is, just by chance, right? I'm going to have more deer exploring soil, right? And I'm going to be able to, right? So that's a risk, right? So the wolves also control kind of the size of that population and make it less likely for outbreaks in the future, right? But I wanted to get to kind of the main point here, right? And it's that we have a structural identifiability issue in the model, right? And so here, right, we have kind of a mathematical definition of what it means to be structurally identifiable, right? I have a model here with P bold and then model with P star, right? And I want to show that Pi is equal to Pi star for all of the for I equals 1 to the number of parameters, right? And so if we imagine very basic version of this, right? If I have a model y equals alpha beta x, right? And this has nothing to do with the data, right? If I have this model, these are my parameters, right? And I write down another version of the model, y equals alpha tilde beta x, right? And then I just do this calculation, I'd have alpha beta x equals alpha tilde beta x, right? X is cancel. And then I have alpha beta equals alpha tilde beta, right? And then so that is not that, right? So I do not have that. That implies the right-hand side, right? Because I have that the product is identifiable here. However, if I just had alpha bar or alpha, maybe I'll do delta and delta bar, right? I would have it, right? So here, I'm going to show you. So I went back into the literature and we were finding like, okay, there is a published value for this epsilon and gamma, but how did they actually do the parameter estimation? Well, they looked across a series of nested ODE models, and I'm just showing you one out of the, I think 5 or 7 they looked at, right? And so the data they had to get this estimate was literally just the density of the dead infected deer, okay? Right, so I don't have any information about the susceptible, the infected, or the prion density, right? But I just have kind of a captive population, and I'm watching an outbreak occur, and I'm counting up the density of the infected, the deer that die of the disease, right? So the data would look like this, right? So I have C here, and then I have time, right? And then it's going to do something like this over the course. But because, right, why don't I know about S and I? This is a really long time course of the disease, and there's no clear way to test an animal without killing it, right? Because I need a brain sample. Okay, so what we show is that, okay, this has that model has this problem, right? You have this unidentifiability. And even if I could track S and I and C, but I had no information about I still have that identifiability issue, right? So we thought a little bit about like, could I measure or could I measure E, what's going on there, right? And so then I talked to even more biologists, right? And then getting samples of E is very, very difficult, right? Because the prion's found in plants, and it's relevant for transmission, right? And it's also found in ticks at levels relevant for transmission, right? So the challenge there, and my friend who's a forester kind of has one of these jobs where you maybe like go out and like take the ticks off moose or something, and then what you find is that there's like 100,000 ticks on a single moose, which is quite a lot. So so those are the challenges, right? The spatial heterogeneity of getting E, samples of E. And then one just last thing about the system, right, is okay, so like when a raven or a wolf, right, eats this infected meat, right, it's gonna pass the meat, excrement, the prions that come through the digestive system and then out again, right? Those are actually still infectious, right? So the one worry is that a bird is going to eat this infected meat and then kind of fly around to a different region and then kind of seed the infection in new regions, right? Okay, and so, right, we do this thing about, wolves could be a good solution to this. But here I was just showing kind of like a proposition from Colorado, where they asked the voters, like, would you like to introduce 30 to 50 wolves over 5 years? And it's very much on the knife's edge of yes versus no, right? And if you've been to Colorado, right, you can see that the blue yeses are in kind of more urban areas or where the ski resorts are, and then the browner regions are kind of more rural areas. So it's a very divisive political issue. Okay, I'm going to switch topics now. And so this project, we're moving into slightly more data, right? Okay. So this one is going to be a gene regulatory network inference problem, right? And so these are nematodes. And the thing to know here is that in terms of their phenotype over the course of development, they can make one of two mouth forms. We have a large mouth form, and then we have a small mouth form. And they develop over the course of about 72 hours, okay? Right, if I have the large mouth form, I can become predatory. I can eat other nematodes. If I have the small mouth form here, I have to eat bacteria, right? So you can imagine it also changes population dynamics a bit, okay? And then here, the task was this, right? So what is known is genetic manipulation to phenotype map. But the biologists don't currently know about the regulatory network structure, right? And so here I'm just showing kind of the amount of data we have, right? So what you can see is that we have about 6 time samples over the course of development, right, which is 72 hours. And we have about 4 replicates, and you can see that there's a lot of noise in the data, right? So if I do nothing and I just let them develop, I get mostly the large mouth form, right? So this is maybe 95% of the population, 5% will be this one. But if I knock out this gene, what I can see is that the dynamics are really different. So if I look at blue here, blue in terms of the mRNA, right, is just kind of going up slightly and then down. If I look blue here, right, it's shooting up and then it's coming back down, right? Whereas orange kind of, more or less does the same thing, right? And then I have one more experiment, right? It also changes the phenotype. That's an important thing to say. I go here, right? If I knock out this gene, I can get back, right? So what we wanted to do, right, it was, okay, so we know genetic manipulation of phenotype map. Regulatory network structure is very unknown. There's a high degree of structural uncertainty in the model, right? So I don't know the regulatory connections, right? And the other thing to say at this point, right, is towards the end of the talk, I'll show you like really cool, amazing data that's you can do like all kinds of cool stuff with. But most of biology kind of lives more in this kind of level of data. And so I think like, there's been a lot of methods development aimed at the kind of the highest end data you could imagine. But why I did this project is I wanted to know, well, okay, like what can I really say if I think about like data that like many people are able to produce, and it's maybe not, as we'll see towards the end, like something that you could kind of deploy like the fanciest methods on. Okay, so I'm going to show you kind of what we did. So where is the structural uncertainty in the model, right? So I'm going to track 3 mRNA, 3 proteins, okay? And there are terms in the model that don't change, right? I know mRNA will degrade, right? I know this term here, right? This is going to be my translation of mRNA to protein. That's always got to be in the model. And then I know proteins will always degrade, right? So what I don't know are these F functions, right? And these are the regulatory connections, right? Okay, so this is saying what proteins upregulate or downregulate each of these mRNA, right?
And sorry, why is the transcription rate the same? Why is it fixed? Like developmental time?
So that's a good question, right? Like that is something that varies over time, but that's not something that you can infer with that level of data, right? So we have to make some assumption, right? So I assume that, okay, the degradation rates of the mRNA are the same, the translation rate is going to be the same, and also the degradation of the protein is the same. Okay.
Excuse me, Cody.
Yeah.
So even if we knew that F1, F2, and F3 were linear, we just don't know the linear weights. Would we be able to figure out what all the parameters are?
And by linear, you mean, yeah, what do you mean by linear?
I mean, F1 is a linear function of E, N, S, K1, and K4.
So I don't know about that. These are going to be nonlinear functions, kind of Michaelis-Menten type functions. We did do a lot of synthetic testing across like a wide range of how many samples do I have, what's the noise on the data, right? And what we found is that if I have incredible data, right, I can recover the true model, but in the biologically relevant regime, what I'm going to be able to say is I'm not going to be able to determine the model. I'm going to be able to determine some features of the model.
Okay.
What do you mean by incredible data? Can you explain what you mean by
Yeah,
that?
sure. So imagine here, right? This is kind of like the standard, right? If I had a way to do maybe like sample the system every 15 to 30 minutes, right? And I could do it with maybe probably 10 replicates, right? Then I'm able to really distinguish features, right?
You just need more refined data.
Yeah.
Okay, thank you.
Exactly.
Do you observe the protein as well as the?
No, and that's the challenge, right? It's very, very expensive to observe the protein, right? So this is another hidden state problem. Okay, so I just want to tell you intuitively, like, what do these FIs do, right? Right. And so what the FIs do is they control, right, whom regulates who, right? And so here I can imagine, like, here I'm showing there's no interconnection, right? But everyone positively upregulates themselves. Here I'm showing a different version where there's no interconnection between the system, but everyone's negatively regulating themselves, right? And then you can imagine there's all kinds of different combinations that can emerge, right? And so for each of the F functions, right, we give ourselves 24 different combinations, and I have 3 of those. And so that leads to about 14,000 models. Okay, and so I'm just going to show you the functional form of what these FI functions look like, right? And so here, right, this is saying like, okay, like the more E I have in the system, right, I'm going to get more and more, it's going to saturate, right? But that the production of the protein will increase as E increases, right? Whereas I can imagine a different system here where the more E I have, the less production I have, right? And you can imagine different combinations, right? Okay, so we go back to this identifiability issue, right? And so we tested just one example. Well, we tested a few examples of the system, right? And what we found is that the model I wrote down is not structurally identifiable, right? And it's because that there is a symmetry transformation in the system, right? And so probably some of us were at Stefano's talk, right, about the replicator equation, right? And he was saying, well, I'm very interested to know all the possible transformations that map the replicator equation back to itself, right? And he gave 2 examples, but the total number of transformations is unknown and something he's curious about, right? So this is actually the same thing, right? This is, notice that if I cancel, right, C everywhere, I get back the same model, right? So that is an example of a symmetry transformation that maps the system back to itself, right? But if I did parameter estimation without fixing this, notice that for any value of C, right, the dynamics would be the same, right? And so the other point, right, is like it's not just going to impact this one model. It's if I add maybe like a more complicated production term here, it's also going to impact that one, right? So we had to kind of correct the model for this structural identifiability issue, right? And it's important to say that this is one we detected. We were not able to screen all possible models. Basically, it's a very computationally intensive part, and it's known that subtle changes to the nonlinearity can increase runtime of the algorithm quite a lot. But we at least made some effort to improve the problem. Okay, so what does it mean? Like, if I get identifiability issues in the system, like we're thinking like, okay, well, hopefully we're trying to shift that more to like the data itself rather than a problem with the model structure. Okay, so now I'm just going to tell you kind of the overview of the approach we took, right? So I don't know the regulatory connections, but I'm going to sample the system, get the data from different perturbations. Then I'm going to fit just thousands and thousands of models, right? Then I'm going to evaluate those fits. And what I'm going to do is I'm going to say, okay, like I'll get some fits that are better, some that are worse. There are practical challenges right? I'm doing kind of like well, first maybe I'll just say like we're doing a maximum likelihood estimation, right? And so some of those are going to be better, some of those are going to be worse, but I'm starting from a finite number of places in parameter space to run the optimization from. So I think we did, 1 or 5,000 seeds, right? But just by chance, right? This may not work perfectly, right? So what we want to do is we want to take those better fits and those worse fits and say, okay, what do they have in common? And based on Becca's question, right? We did a bunch of synthetic testing to know like what do we actually expect in the biologically relevant regime. And what we expect to find there is that I'm going to be able to detect some features of the regulatory network, but not all, right? I don't expect that based on the data I showed you, I should be thinking, okay, there's going to be one answer at the end of it. It's going to be a messier answer of here are some features, right? So I'm going to show you for the wild type result. I do this process, right? And I see, okay, I get this curve where here I have my 14,000 models, and then here I have the cost function value, right? And then here, what I'm showing you is just one replicate. There are 4 replicates for this, but I'm showing you, okay, here's the best fit we got, right? And what you can see is that, okay, it's not perfect, right? But for the most part, right, the simulation is kind of tracing out the data reasonably well. The uncertainty I'm showing you, just one standard deviation here, right? It's not capturing everything, but it looks okay. And then here I'm showing you the worst model we found, right? And what you can see here, especially in orange, right, is right. That model's structure, right? It's not tracing the data at all, and I have a very wide uncertainty, right? So here I take those models in pink and in gray, right? Where I'm saying, okay, those in pink are acceptable fits of the data, and then those in gray, we don't feel so good about, right? We don't think that's a reasonable fit. So then I take those and I just use a simple decision tree, and I say, okay, and I'm trying to classify pink versus gray based on information about the structure of the model, right? So that actually the regulatory network components is a positive, negative, many different things, right? And what we find is we find something like this. So we see like, okay, the things that we can pull out here are, right, 2 of the genes experience positive regulation. There's an auto-regulatory loop for NHR40. There's a positive regulatory connection from NHR40 to UD1. And then there is, and we can't say what type of regulatory connection, but some of the models will have a regulatory connection between NHR40 and this other gene SALT1, but some of them won't, right? Okay. And so here, right, I'm just showing like, here is one of the specific examples from one of the 2 classes we found, right, with all of the regulatory arrows, right? Okay. So then I repeat this process across the experiments, right? And I say, okay, let's play the same game. So I get here, right? You've seen this before. And then I do it for the knockouts, right? And notice that the knockouts, they're going to have way fewer models, right? Because it's no longer a 6-dimensional system, it's a 4-dimensional system, okay? Right? But nevertheless, I can do the same thing, right? And I find the commonalities in the structure there,
And then I ask in what we're calling the model sets in this pink region of acceptable fits, is there a common regulatory motif that could explain all of the data? And what we find that there is, right, and has this like a lot of positive regulation in the system, right? But we're very careful in the paper to say, okay, this is just one possibility, right? And there's a huge amount of uncertainty. Here. Okay. So what do we learn from this, right? It's a difficult data set to use kind of current model selection methods on, right? I have hidden states, I have noisy data, it's infrequently sampled, right? But this is also, this is considered like very high quality data from these experiments, right? So there's this kind of mismatch. It also does a sensitivity analysis over model structure in a very careful way. And I think that's important and maybe not super commonly done, at least in my reading. And it kind of gives you like a barometer of like, what can I actually honestly infer? Right? We do a bunch of careful synthetic testing, right? And there are many caveats that I'm not going to go into here, but I would say just working this low data limit, yeah, there's a lot of choices that one needs to make and be honest about. And almost all the methods I've looked into are going to be better if you have just a huge amount of data. So, yeah. Okay, so now I'm going to shift gears, and we've kind of seen like, I don't have any data, but I have an issue with parameter values. I have some data, right? What can I do if I carefully look over model structure, right? What can I infer, right? And now I'm going to move into a different data regime. Right, so here, this is a problem about physiology of Arctic hibernators, right, and how the physiological organization across different species, right. And I was lucky enough to be invited to the University of Alaska, where they taught me about kind of the physiology of Arctic species and their neurobiology, and then drove me up the Dalton Highway to go to this field site at the very, very top of Alaska. And then this is a day that we were up there, right? And this is like the nicest day of all time in the Arctic or something like that. And it still looks like pretty bleak, but so anyways, I think one thing I got out of it, right, is right, you could experience the environment that these organisms live in and kind of understand kind of the environmental constraints that are important to the evolution of these systems. Okay, so here I'm showing you, this is the body temperature of an Arctic organism, right? So this is the Arctic ground squirrel, right? And here I'm showing you what it does during hibernation, right? And so this is about 8 months of time, right? And then here you can see what it does in the summer. And the summer is about 4 months for them. And in the summer, what they're going to do is they're just going to hold their body temperature fixed. And the high frequency oscillations you see, that's things like I'm running to capture food, or I'm sleeping, right? So I'm going to get some minimal amount of oscillation in the summertime. And then here you can see like I get these massive oscillations in body temperature during the winter, right?
Do they measure this?
How do they measure it? A gruesome process, but they basically like slice open the back of this organism, and then they're going to inject a thermal monitor, and then that's going to send a signal every 5 to 15 minutes, and then there are all these like basically signal receptors in the ground, right? So it's pretty hardcore, but the important part to say, right, is that right, there's nothing else I can measure, right? These are free ranging. Like once they do the surgery, they just let them go for the rest of their life.
Is this one free ranging? I thought this one was carefully temperature controlled.
No, this is free ranging.
How did they get temperature control? Okay. All right. Sorry.
Temperature control.
No, because when I thought, never mind. We can talk about this later. Go ahead.
Okay.
So the data is not uniformly spaced in time, or is it?
It is, yeah.
It is.
Yeah, it's I think it's this one might be every 15 minutes.
Oh, so it's not randomly between 5 to 15. It's one.
Yeah. I think some of the experiments were every 5, right? But then for the newer stuff, but then for the older system, they could right? It's really like the limiting factor is all about the battery, right? Like that's right? Like you can't, that's the problem. Okay, so just give you a sense, right? Like they're doing this for 8 months a year, and then 4 months of a year they're above ground, right? They have this kind of complex societal organization, and it's this example of a circannual behavior, right? And so the University of Alaska is like here, and then like the field site was like up here, like very close to the Arctic Ocean. So it was like a very intense experience. And one thing I learned, right, from like the leaders of that is, well, how do you keep people happy at the end of the earth, right? And it's really about having people be able to eat whatever they want in the dining hall, right, and have like special treats, right? And then that was like a main social driver of like, okay, like I'm not trapped in the middle of nowhere. Like this is an enjoyable thing rather than a hellish thing. So, anyways, in case you're trying to run one of these places, right? And just to show like the differences in the seasonality is like really extreme. Okay, so one long range question in this field, right, is what is setting the spiking behavior? What controls that, right? And why are people interested in this kind of stuff, right? There's a huge amount of money going into these sorts of things, right? Because it's like really relevant to human health in terms of like, these are organisms that are in the summer incredibly fit, right? But in the winter, morbidly obese, right? So there's a lot of interest in like, how does that work? Can we use that for weight loss drugs, right? Another thing that they're interested in is, right, can you impose that sort of like hibernation or dormancy like state in people for long distance space travel, right? So there's a lot of odd things that people are trying to do with these systems. Another thing, right, is here, notice that this is below freezing, and they have a special system in their body where I remove anything from the blood that could be a nucleator, right? And they basically just excrete all that stuff. And so I have a super chilled liquid, right, which is the blood, right? So there's a lot of cool biology happening here.
Take one of these squirrels and slice it open. It would like freeze as soon.
Exactly, and they did that, and it was a horrible experiment. But that was the science paper, right? I cut them, and then I see it, right? The phase transition.
What's if you give us a sense, like, so the ambient temperature of these environments?
Oh yeah. So maybe this is what Neil was asking about. I'm not quite sure, but the ambient temperature is like negative 30 degrees Celsius or something like that.
I looked it up. It is changing, but it's where it's flat is where it's always below freeze. Like it stays below some threshold, the ambient temperature.
Right, but these are like.
But I'm saying like the down dip, right, is before it falls below some threshold.
Yeah.
Like during the fall or whatever.
Yeah, exactly. But it's a like they're outside, like they're free.
Yeah, or free range.
Yeah.
I was misremembering.
Okay. Okay, so there's right, a lot of interest, right? And they want to know like what is the physiological process that's driving that spiking? And there's kind of two thoughts, right? One is on the left, there's a specialized clock that's like the circadian clock, but its period is dependent on body temperature of the organism. Okay. Right, so if I'm here, if I'm warm, I'm oscillating at the normal frequency. But if I'm very, very cold, right, the oscillations are going to slow over time, right? And so the key thing to note here is like if that was the mechanism, I would expect something with a changing period to be driving it. The other one that people have come up with is that, okay, there's something happening. Some system of the body is producing a metabolite, and that metabolite is going to be made only when I'm in the high state. So in the summer, and also when I'm at the very top. So this is like, they do this for like maybe like 8 hours or something like that. And what's going to happen is, go back here, whatever's made is going to degrade, right, while it's in this low state. And then past some physiological threshold, it's going to send a signal that says you have to wake up, or not wake up, but you have to rewarm your body. And if you otherwise, like, you're going to die, basically, right? So those are kind of the two ways people have thought about like what is controlling the spiking rate. And here, I would imagine like that is going to be mostly a fixed period, right? So here, like our goal, right, was we wanted to know like, well, can we use mathematics to kind of figure this part out? And so here I'm having right, this is the first hypothesis. I have something with a variable period. Here I have a constant period, right? And this is a hard problem because I only have access to the body temperature data, right? I know a bunch of these people that are, they have like hundreds of these squirrels, and then they're kind of like taking their blood every hour or something like just a tough line of work maybe. And they're trying to detect like what is the actual thing that causes it. Okay, so we wanted to find a physiological model of this system, right? We wanted to be data driven, right? There were some previous models that had like, I don't know, like 40 parameters, and I don't know, it just seemed kind of, I wanted to do something more data driven, right? And I wanted to be simple and interpretable, and I wanted to tell us something about the biology in terms of like the actual mechanism, right? And then this is a challenging problem because of the hidden field. And also we actually don't know the dimensionality of the system at this point, right? We just know I have body temperature, and that's all we know. So here I'm giving kind of an overview of how we did the model selection, right? So I have the data, right? And we're going to use the blue data here, right? I'm going to save some for validating, right? And I'm going to take the blue data here, and I'm going to do a time delay embedding on it, right? To kind of estimate the dimensionality of the system. And the nice part here is that we found it's actually a very low dimensional system, right? It's a two-dimensional system, right? So now instead of looking for x dot and yi, I'm looking to just x dot and y, right? Okay, and then how I'm going to do this, right? I'm going to start, right? I'm going to assume like a full cubic library for f and g, and then I'm going to chop out terms based on the coefficient size, right? And then we're also using this data assimilation inspired cost function where I have a data error, then my model error, right? And I'm basically just sweeping over beta, which is how much do I weight the model enforcement versus the data error, right? And also how big of terms should I chop out based on their coefficient size, right? So we go through this process, a computationally intensive process, and basically we look along the Pareto front here that balance. We're looking for models that are simple because I want it to be interpretable, but also give a good fit to the data. Right, and what we see along the Pareto front is I get models with a conserved structure. So I'll just say here, like basically we get models, and the thing that's varying along the Pareto front is just whether they have these pink terms in them, right?
So is the realization this is a low dimensional system from the outset just sort of specified by there is a really regular period, like there's a really regular periodic structure with the same amplitude every time? What is the content of your earlier statement where you were like, oh, you didn't need plurality of y's, you only had one?
Okay, so to do that, right, like what we did is just, right, if I have, maybe I'll just show exactly what we did, maybe easier. Okay, so I have, I want to estimate the dimensionality of the system, right? I've got my time series x1i, right? And I want to know the phase space that it's coming from, right? So I need to sample both the lag and also the dimension of the system, right? And so I'm going to do this process where I'm going to go through and try to right, find those, right? And so often used for chaotic systems, right, to estimate the dimension. So that's kind of like a mathematical answer to like what we actually did, but there's actually like more like a model-based answer, right? Which is like, okay, if we say it's 2, right? And then we go through this process, and then we look at the model, like, does it work or not, right? Maybe a couple other things to mention here, right, is like we did some by hand stuff, right? It didn't work perfectly, right? So like we were chopping out like terms based on the coefficient size, right? But this coefficient, this theta naught is like really, really small, right? It's like 1 e to the minus 4 or something like that. So it's super small. So I had to put that one back in by hand. And here we also looked at like not just what is the coefficient size, but how big is the like theta 1x squared or theta 2x cubed during one of these spikes. And what we see here is that if I look at just the term size alone, some of them are really small. They're kind of inconsequential, right? So these terms here, like these cubic terms like y cubed and xy squared, we actually don't really need them. And what we show later is that they introduce features to the model that we actually don't really want. So we do super high dimensional model selection, then I get the model structures, and then I'm going to just refit in the lower dimensional spaces, right? So my base model is just going to be the black terms, and then I'm going to add in, blue or or pink, right? I'll just add in 1. That will give me 4 combinations. If I add 2 of those, I get 8 and so forth, right? And then I'm going to fix one of the parameters for identifiability reasons, right? So I go through and do this, right? We fit to the training data here, and then we do the out of sample data. We're going to do like a dynamic time warping, right? And so here I'm showing you the fit of the X state to the data for the simplest model. Okay, that looks pretty good. And then here we were wondering, like, what do those more complicated terms do in the model? And what they actually do is they introduce a like a refractory period after the spike, and that's not seen in the data, so we don't like that. So we went ahead with the simplest model, right? And then here, just to get back to the original question, right? I'm showing Y in the dashed pink here, right? And I see like, okay, like I'm gonna see like probably it's gonna have a constant period. But an important thing to note about the biology here is that so this interpretation of Y is saying something about like, okay, like something is building up and then it's being depleted in the low state. But I can also actually rewrite Y, because Y is not being fit at all, right? So I can say if I have Y tilde equals minus Y minus alpha, right? Then I can flip that state, right? And so that's what I did here. It doesn't change the fit to the X state at all, right? Because it's right. This is an unknown or hidden field. And so what's happening is now this interpretation is saying, okay, it's not something depleting, right? It's something that's building up. A toxin is building up, right? And past some threshold here, then I have to rewarm, right? So we're careful to just let people know, because people are looking for these things, and that's actually like a pretty different dynamic.
Cody, if I understand Y to minus Y I can do, but if I add the minus alpha, do I not change the sparsity structure model?
I don't think so if you pick it carefully, but I'll have to check, right? Okay, and so, right, I told you it was kind of this very interesting circannual behavior, right? And well, anyways, it was just something I wanted to think about, kind of the neural mechanisms a bit more, right? Because these circannual rhythms, one of my scientific heroes, Winner, this German ornithologist type person, right? He would keep his birds in a lab in a constant environment for 12 years. And what he showed is that even in constant environments, every 11 months, they exhibit a flapping, right? They want to leave for migration, right? So it's an endogenous process, right? That has some interaction with other environmental entrainment, right? But we know so much about circadian biology, right? And this is kind of like a year version of that, right? And it's seen kind of all over, right? In hibernation, migration, coat changes in hare and fox. And anyway, some newer work is showing that you have these oscillations over the year scale in human endocrinology. And so I thought, okay, like, you know, could I find something like that in the model? Could I get to something where it's closer to this multi-scale process? Right? And so here, what I'm doing is I non-dimensionalize the system, right? And so then we're gonna carefully see that each of the parameters is quite interpretable in terms of what it's doing to the actual biology, right? I look at this hop curve again, right? And what I see is that if I look in gamma and then XC, right, I get kind of different regions, right? So in this region, I have oscillation, right? That corresponds to the winter behavior. And then here I have the stable summer behavior, right? And then the neurobiological input of the circannualism, right, is the shift in that parameter potentially, right? And so I'm just showing like this is a trajectory of gamma such that you get, right, the broad features of the model. And it's not perfect, right? I'm seeing more like, right, I don't have those kind of like kind of noisy trajectories as I descend into hibernation, but it shows the broad features of the system. And so in terms of like what everything's doing, right, this is the seasonal tie-in from the neurobiology to the physiology, right? And then here, see what it's doing is if I change C, right, it pushes the amplitude down, which is another thing that they see that the organisms will do if I have a hibernator in a very warm environment. It will still do the behavior, but it will lower amplitude, right? And then And then these two parameters, iota and kappa, right? Those are related to the internal like physiological functioning of the either accumulation or depletion of this metabolite in the physiological threshold. So then just quickly, right, I knew a lot of biologists and they were all measuring these body temperatures, right? And I wanted to say like, okay, we have the one model we discovered here. I do the same dimensionality reduction, I estimate the dimensionality of the system and I see they're pretty low across organisms. If I take my model and then I add some environmental information to it, can I qualitatively get the same, can I get something that looks like this, and that? And the answer is we could. All right, so these ones, the shrew and then the bird, they live, they have access to photoperiodic information, right? But they're still changing their body temperature, right? So we add something that looks like a circadian signal. Here, the bear is hibernating, right? But it has this kind of behavior, right? So I add something that's not circadian, but maybe like longer wave information, something more circannual. Okay, so because we were running out of time, maybe I'll just, right, that's kind of what we did. We developed a model. We were able to distinguish one hypothesis from another in some sense, or at least found support for one. Find model that captures broad seasonal transitions, right? And then we start to think about like how our physiology of different organisms organized across species, right? Okay, and then I just want to show you the data. I won't tell you about the whole story, but this is like a really heavy data regime. Okay, so this is data I got from the city, and I FOIA'd them to get this. Big battle with the rat czar and all this stuff, but basically, like you're seeing these spatio-temporal dynamics, right? Of like, who is complaining about, can you come to my house and kill the rats or whatever? And then here, right, is a graffiti removal. Someone has spray painted my house, and I want that to not be the case. Where is that occurring in the city? And then here, right, the weed removal. This is, I have a vacant lot near my house and clean it up. And then the last one, right, more global responses, right, of like snow removal and things that impact everyone, right? So we're starting to see, right, kind of interesting dynamics. We're doing a bunch of work to like kind of get towards like mechanistic and dynamical systems understanding of city dynamics, but there's no time for all this. So I'll just thank everyone that helped me and the funders. And I'm happy to take questions if people have them.
Super, super cool. So in your cross-species comparison of the temperature regulation, you kind of come to some sort of speculation about whether this one mechanism is happening for all species, or whether there's evidence that like one species uses the other mechanism.
So what we show is that there's or what we suggest, right, is that across species, right, it can be different, but the organization is actually quite similar, right? And you can imagine like here, I don't have to have any information, right, from the outside world to have the process occur. But if I have that same systems organization of the physiology for these other organisms, and I add environmental information, it can still, the core mechanism can occur.
I'm curious if you did a comparison or looked at data for insects that also have circadian rhythms, and same with bacteria. So there's certain bacteria that have protein receptors that show the circadian clock, and I'm curious if.
So the only thing I looked into regarding that is the honeybees will have this kind of behavior where they modulate their body temperature quite a lot. So I haven't done that, but I'm hoping to look into that.
Yeah, praying mantis do this.
Oh, okay, that's very cool. I didn't know that.
In your model, you show, I think if you look at the data, there's a little bit of period variability across the spiking, right?
For this one?
For the model, for the organism you fit for the squirrel.
Yeah.
Have you thought about making, everything you do here is deterministic, if I understand?
Yeah, that's right.
Have you thought about adding some sort of stochasticity to the model to try and replicate some of that periodic variability? And maybe that would give the noise, it becomes more sensitive to noise when it's high temperature and less sensitive when it's
Yeah, I haven't done that yet, but it's something I would like to do, right? To kind of capture this part of the dynamics of it better. But also, like you were right that, and this was actually a very annoying part of the fitting was it's like pretty much the same, but it's not like one might be like 8.5 days, and the other's like 8.7 days, or right? Like it's so like it's not perfectly periodic, and it's like an intrinsic noise that's causing it in the system.
Let's thank Cody again.
Thanks.